Twin Peaks. You know it's time to get the lab coat on. Twin Peaks. Yeah. Cause we out here being hella wrong. Twin Peaks. And you know we're never stopping till the hammer drop.
Welcome back to Beak, where we make you compliant with science and the techno capital machine that is coming for your lives. What do we
It's
got
got a
today?
blicky. It's got a blicky pointed right at your forehead, and if you don't understand what Technocapital is going to do with that blicky, then I guess, ignorance is bliss or whatever. We've got a wonderful article today, Salty Squid. It's by the name of Four-Dimensional Molecular Mapping from Spatial Snapshot Reveals the Dynamics of Hair Follicle Organogenesis. Now, this title is a misnomer because it is not about hair follicle organogenesis. This is a marketing product. This lab, straight out of the Johns Hopkins, they did nothing to do with follicle organogenesis. They're promoting a product. Now, what product are they promoting? Let's take a look at the abstract.
So, the abstract, or what they're calling the summary here, says, understanding organ formation requires capturing molecular information simultaneously in three-dimensional space and across developmental time. To this end, we developed 3D DNA-enhanced expression profiling, 3DEEP, a tissue-clearing approach that removes genomic DNA to extend spatial transcriptomic profiling hundreds of microns into intact tissues. We applied 3DEEP to neonatal mouse skin, capturing hundreds of developing hair follicles across their organogenesis trajectory. Ordering follicles by molecularly inferred developmental age transformed this single spatial snapshot into a four-dimensional, 3D plus time, molecular map of organogenesis. This map revealed developmental dynamics spanning stem cell compartment stratification, emergence of new cell subtypes within the follicle, and cascading structural transformations leading to hair canal formation. Comparative analysis of FOXN1 deficient nude mice, a hairlessness model, revealed organ-wide changes in developmental dynamics, including delayed molecular progression, reduced coordination, and increased developmental instability, preceding overt structural defects. This work demonstrates how deep tissue spatial transcriptomics can uncover hidden dynamics of organ formation.
Yes. We love tissue clearing. We love spatial transcriptomics, two of the coolest recent developments in science. But spatial clearing has been sort of limited to neuron tissue, mostly because clearing a section of neuron tissue can give you a lot of information about the spatial organization of cells within it. But for tissues besides neurons, outside of these very fatty tissues, there's a lot of challenges. Now, spatial transcriptomics, also insane technology where you take a slice of tissue, you put it onto a glass slide, and then basically you can take images of the tissue and find out at a single cell resolution what these cells are expressing, like what sort of mRNA are they putting out.
Yeah,
And the big
but
thing
for this one,
in
I was going to say, for this one, the real important part is that they're taking it from neonatal mice, and that allows them to get a snapshot of all of the follicle formation at all of its different progression points, because they have some hairs that are, just newly forming, and they also have hairs that have been forming for a while.
Yup. And so they mention organ-wide changes in developmental dynamics, delayed molecular progression, reduced coordination, and increased developmental instability. All the big things in organ development right now, they're sort of pointing towards it's not just having the right concoction of genes, it's the timing of these genes.
When they're being
If you don't
expressed.
have Yeah, they need to be expressed in the right order at the right time with the right oscillations. Maybe we'll do like another paper in the future about like expression oscillations, but that's the big thing in
I think
organ
we just need to
development.
do a
But
baseline how genes are expressed via, DNA transcription and all that.
that would be a good show, yeah. But there have been quite a few developments since the standard model of genomics. We've passed it. It might be useless, but it's still going to be taught in like high school and everything. It's like gender. We go from DNA to mRNA to protein, just like we only have men and women. Pretty much. This is third grade biology. But this is not about organ development. This is a
Yes,
marketing
but
product.
Dispel, do you know how many people in America don't understand biology up to a third grade level?
They probably wouldn't be able to tell you the difference between DNA and RNA, but they could certainly tell you what a man and a woman is.
Anyway, figure 1,
And men are useless women, is what they are.
men are useless
Is I
women.
think is what I think the consensus has been with the right wing.
Oh no.
Okay, on to the 3 deep technology. Why are they talking about DNases? So,
I
like I
was
mentioned,
3
tissue
deep,
clearing.
3 tentacles deep in your mama last night. No, I'm just kidding.
I've been refraining from making this joke the entire time, and I hate that you've brought this platform so low to make this joke. But I'm glad it wasn't me. I'm blameless. I'm entirely blameless
Well,
because all I want to do
you've
is
taken
talk
a lot
about
of
tissue
heat
clearing.
lately, so I was trying to take some for myself.
All I want to do is talk about tissue clearing and why tissue clearing is easier in fatty tissues compared to something like a skin tissue, which is also an organ, by the way. Basically, most tissue clearing has been done in fatty tissues because you can just throw a bunch of solvents, you can throw a bunch of detergents at fatty tissues, wash away all of the fats, and you'll leave behind essentially a matrix of protein, which you can then stain with fluorescent antibodies and then image in different voxels. But for more protein-rich tissues, it's a little bit hard to get your imaging stuff in there. And so these people out of Johns Hopkins, very clever people, they believed that all of the DNA that's accumulating in the keratinocytes is sequestering the reagents from truly penetrating the skin. So if you take a look at figure 1B, they've got a nice little diagram on why the DNase is important to this product. On the left, they've got tissue treated without DNase, and you can see that it is very, very blue. That blue represents DAPI, which stands for nuclei. On the right, you've got tissue cleared and treated with DNase. So all of the DNA specifically was broken up and chewed up and washed out. And you can see that it's primarily red, which is a apoliptin protein. The good kind of cholesterol is what it tags for. And so this is just saying, the DNase works great in allowing us to image not neurons, excuse me, organs that we wouldn't normally be able to do. So if you take a look at D and E, they've got exactly, oh my God, they've got literally the stars are under your skin. I bet you didn't know that.
It's beautiful. It looks like a Doctor Strange multiverse of madness parallax. I love it.
You're made of stars. It's true.
You are
But
Stardust.
it's true. You are Stardust. But the idea in like the ontology of biology right now is that cells are what they express, and you can identify what a cell is not from histology, but by working backwards from what it expresses. And there's going to be all sort of cacophony of a specific cell type that is expressing a particular protein. And that's what they're doing. So they got oligodendrocytes, GAD1 corresponds to GABA neurons, SLC 17A7, glutaminergic neurons.
But
SLC 17 is probably an NA voltage
what's the
gated
point,
channel.
Dispel? Why do we want to map out these follicles and their organogenesis and see them from point A to point Z of their development? What's the point? What does that help us do?
That's a great question. Well, if we're going to be growing organs in the lab, we want to make sure that they're biologically relevant and they're developing properly, and that we can troubleshoot any organs that we grow in a petri dish for your cirrhotic liver because
I
you've
saw
been
somebody
drinking too
post
much.
about some Yahoo on Twitter the other day that claims that some company has built him a petri dish baby for him to harvest organs from. What do you think of that? Is that technology out there on the dark web?
On the dark web, no. On the dark web, that would probably be like actually developed people's organs. That's probably what.
True, 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 true.
So actually, it would be far more moral if we could do it in a petri dish. That would be so awesome,
Is it gonna be like
Even
the what 2005 movie, The Island? Is it gonna be like The Island where it's Ewan McGregor as a clone trying to escape the underground facility?
no, see, like, yeah, the problem is like when you want to develop like the entire thing instead of just the organ.
Yeah,
That's
can we
kind of
just can we just express the genes that make my hip and then implant a new hip in me?
that would be cool. Yeah,
Like
we
just
might be able to do that.
3D print me a new hip with my genes, you know?
Yeah, we might be able to do that. That would be cool. Honestly, it might be like nanobots. But let's take a look at what they were up to. Introduction, whatever. We gathered a bunch of transcripts from 220 genes across 400,000 cells that they isolated from neonatal mouse skin. So yeah, this is the sort of thing that the right wingers are upset about with harvesting babies
Oh
and
no,
abortion.
I thought animal rights was like a left wing thing. I thought the freaks that were blowing up animal research centers were like on the left. Am I wrong about that? Are they right wing
Well,
extremists?
no, 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 no. See, like I'm so bigoted against animals that I don't even like think about that, and I immediately jump to how can we apply this to humans, and who would be upset about bringing this to humans because
How
I am a
can you be
progressive
bigoted
at the end
against
of the day.
animals? You are an animal.
Not an animal like you. Who?
Oh my god. Out of the two of us, which one of us is more likely to be found in a lab testing scenario? A squid or a pigeon? Yeah, that's what I fucking
I
thought.
think
No, that's what I
I
thought.
think the pigeon days of research are over. Like we already guided like bombs. They tried, failed. I think it's maybe the time for the cephalopods to get a little bit of work done on them, you know?
No, I don't want to be cut open. I don't want to be a lab freak.
Well, maybe a little bit of pushing, a little bit of prodding. But let's take a look at figure 2. Uh we got figure A, which is just, what they extracted out of the skin. We've got the three-dimensional voxels that they reconstructed from that. I don't know how big these voxels are. That would be a very good question. But then they essentially grouped together the signal into presumed cell types and then built a model of the hair follicles from each of these little things.
Okay,
The
so
big thing, salty squid. Take a look down here.
yeah,
What do we
F.
got this.
F is the
We've
meat
got
and
like a
potatoes.
magazine. We've got a factory down here in figure 2F. Transcriptomic capture and spatial pseudotiming reconstruct hair follicle development.
This seems
This
like
is
this
the
was the
hair
whole
follicle
point.
factory. Yes, 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 yes.
The
So
reason they
they
created
have.
this technology, the 3D technology, so they could do shit like this, is where they can map out from the youngest to the oldest the cell's development and organogenesis. And this is going to be very helpful for understanding like how disease progression happens, how we can fight disease, because we can't fight diseases that we don't understand. And the first way to understand a disease is understanding the organ that it affects.
Well, I guess the alternative is, also just like letting people die. That's kind of how we did it for a couple
One of the
hundred
interesting
thousand
things
years.
that they found in this article that they didn't know before is that during organogenesis of the follicle, they thought that the where the shaft is, they thought that that came from the bottom and that the hair follicle was pushing out. But in fact, two pores happen at the same time, one at the surface of the skin and one below the skin, and they meet in the middle to create the shaft where the hair follicle will be.
Ew,
That's kind of interesting.
ew. That is interesting. Like, there's got to be some sort of like coordination between the top and the bottom.
Yeah, they
Like,
they
how do you
coordinate and they
do they
meet in
just
the
line it up?
middle. It's very
Uh-huh.
interesting because, I mean, intuitively, you would think that no, you just have a follicle form beneath the skin and then it
Like a
pushes its
seed.
way out. Yeah, like a seed.
Yeah, like a
But
seed.
that's not actually how it occurs. It actually creates two different pores and then they meet in the middle. Fascinating stuff.
Ew. Ew. Like
Listen, that's
you
something
hair
that I
growers
learned
are so
today,
gross.
and you should be proud that you are better than you were yesterday, that you now know this.
I'm just marveling at this like figure 2F
Oh,
and
it's
like the
gorgeous.
factory. Well, because like the thing is, I don't see how they're like meeting up against one another. I don't see where the like the hair follicle is until like you get to like these bottom three rows where like, yeah, that looks like a hair follicle.
Because I think that it's showing the expression of the genes, which would happen from the middle outward if you're having the pores meet in the middle, right?
Hmm.
So there's a
So
point
like,
in between those genes because it would have to have a way of those 2 points knowing how to connect to each other perfectly, and the only way to have that happen is for the genes to be expressing from the middle of those 2 points.
it is crazy that you have the audacity to look me in the eye, in the beak, and tell me that God doesn't exist, and you feel astonished that biology is regulated like this.
I mean, we don't got to bring God into this. He's got nothing to do with it.
Speak your piece, atheist. Hmm? Oh, just random chance. Go ahead, do it. Do the thing. I'm an atheist and I hate a religion and I think that all of this is incredibly interesting and miraculous, but no, it couldn't have been designed by an all-knowing creator beyond our comprehension.
I think it is
It's just
all that
random.
more miraculous that it happened by chance than if it was meant to be.
Yeah, well, maybe you should start rolling like a couple of d20s. Hmm?
Hmm? Hmm?
If you think you're so lucky. Mhm.
We
I'm
should
pressing
we should do an episode on
text.
religion. That'd be fun. We both have a lot to say about it.
Oh, this is our episode on religion.
Oh, this is our
And
episode on
I think you're
religion.
failing dramatically.
You dumbfounded me to come into a discussion on religion. Well, I don't want to hurt your feelings, my bird brain friend. You're allowed to have your
I've
religion.
I have rug pulled you like 3 times in regard to this episode. Because first I told you this was a balding episode. Then we
We
find
haven't
out
talked about
actually it's
Tim
a
Pool
marketing
once.
episode. No.
I
What does
thought I
Tim
was
Pool
coming
have to do with
here.
balding? He
What
was
do you
born
mean?
that way.
He was born with a beanie attached to his head?
Yeah.
I guess that makes sense. I was also born with a hat attached to my head.
We're gonna have to do some tissue clearing on Tim Pool's skull. And we're gonna find out that there's no brain between his ears.
There ain't enough tissue there, brother. It's all skull. It's thick as
No.
fuck.
No, it's actually all beanie yarn.
You
It gets replaced by like
just start
shark teeth.
unraveling his whole head and he'll just go undone.
Yeah, like people think that he's got like hair in his ears. No, that's that's a yarn poking out. And
Goodness.
if like you keep tugging, he's gonna run out of beanie.
Yeah, I hope Timble doesn't watch this episode of Twin Peaks at all, ever, because the organogenesis of hair follicles, that's a touchy subject for him, I'm sure.
Yeah, but it's like this is about like newborn organogenesis, not like
I mean,
mid-born.
he seems like he was born yesterday.
He was born mid as well.
He just came out a
But yeah, based on the
full
way that he talks politics,
grown man, bald as fuck, and they slapped that beanie on him, and now we're cursed with him.
God, that sucks.
All right, on with the article. That's enough shenanigans.
I don't know. Like, I feel like we explained most of it already. And because like the pictures are just so pretty. I'm parsing through the text pretty quickly, and it's like, yeah, you know, RNA sequencing to confirm this, and these are the proteins that we use. That we already see the proteins in Figure 2F. Yada yada yada. I don't know. Like the stars under your skin are just more appealing to look at than like the words on the page.
Oh, so you're a figures guy? You're a big figures guy? Have you seen Figure 3? Figure 3 is also beautiful. Figure 3D, funny enough, 3 deep, shows a picture of the hair follicle and all of its structures. And a couple of things they learned from this is they didn't know like a couple of these structures even existed before they did this study. So pretty cool that they're learning more about how hair follicles are formed. Because again, the more we know about an organ, the better we are to fight diseases that are associated with it.
Yeah, like baldness.
Not
Alopecia
everything is
actually is probably
baldness.
it. Alopecia.
We could have saved Will
Oh, what?
Smith's marriage if we just had the cure for alopecia, and this is the kind of
I think he
technology.
likes the abuse.
Chris
Am I
Rock,
allowed to say
listen,
that?
Chris Rock would have never got slapped. He would have never got personally traumatized in live 4K if we had saved Will Smith's marriage by curing alopecia if we had this technology earlier. So that's what I'm saying. We're on our way to saving marriages in Hollywood.
That's so cool. We love that. We love that for
Because Jada Pinkett
marriages in
Smith does
Hollywood.
have alopecia, in case you didn't know.
Oh, wait, they were both
No,
bald. That's
yeah,
right.
we can show that bald ass head. She's not a nice lady.
Wait, you think that she would have chosen someone else if she had hair?
No, I think she would have not cucked Will Smith. You can't
But
do
what
that
if
to
he
the
likes
Fresh
that?
Prince. He does
Wait,
not like that. That man
why
be
can't?
crying. You know it.
Yeah, but what if he? Is it not okay for men to cry and like getting cucked?
Um,
Maybe you're just coming at this from such a like a cuck-phobic mindset, Salty Squid.
I do have a very cuck-phobic mindset. I'm not gonna lie. I don't think anybody should be cucking themselves. Please stop. Have some fucking respect.
Okay, we're locking into figure 3. Is what we're locking into.
I'm locked in.
Yeah, I don't know. Is there much to talk about in this article? But like, be amazed at the figures that they've presented because the figures are like relatively like we've sort of explained what's going on, the
Yeah, but
premise behind it,
if you
and
look
like,
at Figure 3D, you can see what I'm talking about when they discovered that the two pores generate and then meet in the middle because it's got kind of a bulbous part here near the pore surface and it's got a bulbous part here deeper below the skin where the matrix is, and so those two areas form independently and then meet somewhere in the middle here to create the full follicle shaft.
mm,
Very
mm,
interesting
mm,
stuff.
mm.
That might be the most important thing that we learned from this.
I mean, honestly, like the insane thing about the hair follicle is just the number of layers of different skin types that come together to what essentially looks like a flower, in all honesty.
Well, honestly, the one limitation, well, probably not the one limitation, but one very serious limitation of the study is that they did use mouse hair, which is not applicable generally to all different types of hair. Human skin has different products in it that melanocytes that leak in and pigmentate your hair follicles, so it gets your hair from like a blondish color, and that's what pigments it to like brown. So this study doesn't include any of that. I think it'd be interesting if they could do this to humans, but I think that would require slicing off some of their skin, which I guess is not ethically viable.
I don't know. It's like a little hole punch. Like,
I feel like there's
I
some
feel like
people that would allow
it's
that.
probably okay. Yeah, I feel like it's okay.
So yeah, I guess they started with mice here, but I'd be interested to see, other types of skin, other mammals, even non-mammals, what they can do with this technology, just to get a good database ready for when they use it for its real function, which is to, scan other organs and figure out how to prevent disease. Is the ultimate goal of this tool.
Oh my God, Salty Squid. I completely forgot. They've got a website. They've got an interactive interface for the map. Show me the money. Show me all 64 megabytes of money. Color by. Holy shit. And then you at the bottom you can see, oh, they ranked them by time. I
Yeah, so
see.
this
So
is just the
you can pull it
progression of the
across.
expression of each of the genes that they colored.
Right, right.
And you can see it's
Filter.
spreading out from the middle to the top and to the bottom, which
Okay,
is
you
kind of
can
like what I was saying.
change the
That's how
cell
the two different
types.
pores form.
Yeah, it
Colored
looks like
by
it's sort of an
cell type.
involution from the top down.
Yeah.
Actually, color by structure. There's not much in this user interface, actually. It is cool, though.
It's cool to be able to see the progression, but it's not you're not seeing the hair follicle be built. You're just seeing the different genes that build it. The
Correct.
fibroblast being
Which we do
expressed,
presume to be the cells.
which
Data loaded 30%?
you can see down here in the left corner, they've got the different types of cells that are being formed. You got fibroblasts, which would be the genesis cells for fibers of like hair fiber. Keratinocytes, which are going to be what's making up your skin, or keratin, and then melanocytes, which are going to be the coloring agents and the pigmenting agents in your skin.
Oh my god, salty squid. Up
Yes,
top
ma'am.
is the wild type hair.
Uh-huh.
Down below is the nude mouse hair follicle.
Okay.
Why don't you chew on that for a moment?
Why are we
So
comparing the nude mouse to the wild type?
to compare and contrast the development of the
Well, you can see
hair
less.
follicle
You can
over
see.
time.
Well, I know some spots you can see less expression in the nude mouse, but at other points it looks like it's expressing more genes.
It looks like the like knockout mouse tends to like reach deeper into the skin at like random points in time,
Yeah, but look
which goes along
at
with their conclusion that there's some sort of coordination that's going wrong.
time rank 260.
260.
It only goes up to
Okay.
262. Look how much bigger that is on the wild type. At
260.
the end, it just fucking goes
Holy
crazy.
moly. Yeah, okay. Wait. Yeah. But okay, like, I mean, the sample 258, that one looks longer than the one above.
Sure.
Yeah.
Okay.
Yeah, I don't know. By cell type, they don't look
Okay.
too different.
I'm looking at it colored by gene, and then you can look over down in the left-hand corner and see each of the genes that's being expressed.
All the 226 genes that they're expressing.
Yeah, which you can, associate to each of these colors. That's pretty cool. I'm not going to
Holy
take
shit.
time to figure out what each of these means, but they have something to do with building a hair follicle. I know that much.
True, but I can't tell, like,
This
by
little one here is
eye
saying, please build a
makes a difference.
fibroblast. Yay! This one over here, it's saying, you're going to be a shaft. Yay! And then this purple one over here is, you're going to be a matrix. Yay! And then that's how shit happens. Biology's beautiful.
Oh my god, I was right. Okay, so the authors say, our 4D map also resolves important details about hair follicle organogenesis. For example, based on histological staining, the matrix is thought to generate the IRS well before the hair shaft, suggesting a well-formed IRS may be necessary for hair shaft induction. I mentioned the IRS. Oh, not that they're investigating me.
Dispell.
I mean the 3D map.
If you go to the legend, you can randomize the colors of all the genes, and you can just keep clicking that and give yourself a little like twitchy rave.
It's very pretty.
I hope we don't have any epileptic viewers because I'm fucking them up right now.
Oh, avert your gaze. So they continue saying,
No, we love the
unexpectedly.
gaze. Don't avert the gaze.
Okay. But so they say, hold on. Unexpectedly, you keep fiddling with that. Our molecular map shows that the IRS. Wait, hold on. Can you stop fiddling with it for a moment and
No,
find
it's
the...
so ADHD friendly. Look it, I'm
Go
a
to
master of the
structure.
dot.
I'm a superhero! I'm a motherfucking super...
Structure.
Okay.
Color by structure. Color by structure.
Okay.
Find your IRS color.
IRS. It's yellow. It's fucking everywhere. Holy shit.
Make it stand out. And then I want you to scroll through the pseudo time while I describe.
Why is the wild type stream so much thicker with beautiful rainbow colors of genetics than the nude mouses?
I wonder why the normal acting hair is like that and the nude is not. That's so curious, salty squid.
I'm a curious guy.
This is the first time that I've actually like demeaned you for making an observation, which is not something we should do in science. I'm sorry.
I love making obvious observations. What do you mean?
Well, I mean, the thing is, just if it's obvious doesn't necessarily mean that it's actually obvious. You know what I'm saying? Our molecular map, bald yourself, motherfucker.
Why? Okay, IRS. Why is IRS important other than they're trying to take my money?
So they say our molecular map shows that the IRS and hair shaft emerge concurrently between pseudo-time 0.35 and 0.55 during the second molecular stage, with their cell numbers highly correlated throughout organogenesis. Consistently, IRS markers and the hair shaft marker S100A3 begin to appear after pseudo-time 0.35 in distinct cell populations at the base of follicles. By pseudo-time 55,
Follicles? What's a follicle? Is that like a follicle that goes wrong? Follicle?
foul. It's a pun for like birds. Follicle.
Oh, okay. Is that where feathers come out of?
Correct. But that's not what they said here. This was rats. Disgusting.
I know a couple of
Marsupials.
rats.
By pseudo-time
Rats are not
0.55,
marsupials, motherfucker. What?
The stacking of IRS above the hair shaft is clear in all follicles, and the IRS begins to acquire its characteristic conical shape, nestling the hair shaft inside. So the
Okay.
authors analyzed the distribution of dendritic cells and macrophages, which are sort of the resident sentinels, within 10 micrometers of hair follicles over pseudotime. They found dendritic cells did not exhibit depth-dependent bias along follicle length, consistent with their uniform distribution in the skin. Macrophages showed a different pattern. They were predominantly located in the lower dermis with a minor population beneath the dermis. Follicles grow nearly downward until approximately pseudotime 0.7, reaching roughly 90% of their maximum depth before beginning to bend horizontally. Consequently, they encounter two distinct macrophage density zones. The first, early in organogenesis, the follicle resides in the low macrophage zone. As it reaches approximately 125 micrometers deep, its lower portion encounters the high macrophage zone, or figure 4G. Where's figure 4G? Oh my god. That's your high macrophage zone? Okay, sure, whatever. It looks like a modeling of macrophages. This encounter at pseudotime 0.3 coincides with the transition to the second molecular stage and bulb formation, raising the possibility that microenvironmental cues contribute to activation of the second molecular program. It could also be you hit a different stage of tissue, and that's sort of the threshold for it to start bending.
That's interesting. So the hair follicle shaft is not a straight up and down shaft. It's kind of curved at some points.
It seems like it gets curved at some point, yes. So it starts straight up and down, orthogonal, perpendicular to the surface of the skin, and then it curves in a particular direction.
I mean, then that's even more impressive that the two pores can find a way to meet each other in 3D space.
Yo, you know, it'd be an awesome like investigation of this sort of like macrophage zone is the development of like cowlicks. You know how people will have like
Oh, bro, I got a horrible
spirals
cowlick
in their
under this
hair
hat.
with like yeah. But like that's a spiral, bro. How do you get like a gradient that is like pushing all of the hair follicles in a circular pattern in a particular way that gets disrupted at a certain point? You
It's
know,
a
as you get to like
Fibonacci
the rest
sequence,
of your
bro.
maybe, yeah. I believe in
It's
the
a
natural
perfect
numbers.
spiral in my head. I was touched
There
by the
are-
angels.
You were certainly touched
The
by
demons.
you wish. Okay, what do they got in figure 4 over the entire thing? Yep. Lots of things going on over time that they use to define the stages.
I mean, yeah, the stages, as you can see, are not all equal in their amount of expression of genes. You know, some of these stages, they're just kind of chilling. They're not really expressing, not growing, not doing things. It's kind of a middling stage before they get pumped up to express a bunch of genes and grow at once.
Yeah. So like the episupra, the basal, all sort of like are level. The epidermal melanocytes, they're like the number of cells there is relatively even. But then as you get into the later stages, your CP, your IRS, your fibroblast, your matrix, your hair shaft, all of that starts to accelerate.
Beautiful coloring on these figures. It's very ADHD friendly.
It is. Is ADHD, just accepting that your childlike wonder,
I think so.
never losing your
You
childlike
have to
wonder?
you have to give in to the ADHD and the chaos. You can't fight it.
It might be, yeah. Okay, but it looks like, I mean, their molecular stage of organogenesis in figure 4C looks like it describes exactly what you're talking about. It looks like the melanocytes sort of start involuting and they meet a layer of stem cells that starts to bulge outwards, where the upper layers start to develop into the upper cavity and the ostium, and the lower layers bulge out into the lower cavity.
Yeah, fascinating stuff.
Yeah. See?
Yeah, see, this is how hair grows. We thought that it maybe grew like a stem right out of the fucking skin, but no, they meet in the middle, those two paws.
That was really good. Oh my god, that was really well done. Okay, then I don't know. I feel like is this it? They've got
That's
figure
the meat and
5.
potatoes. The meat and potatoes is we learned that hair follicle organogenesis is not what we thought it was, and the meat is that we have the 3 deep technology that we can use on not only hair follicles of rats and mice, but on hair follicles of other animals and on other organs to discover great new things that we don't know about the fucking organs. I don't know, man.
Oh, okay. I found this 2 cavity model of organogenesis that they're proposing, because I don't know what the fuck you were talking about.
Well,
I
you
think I'm seeing it.
clearly didn't read as much of the paper as I read then.
I was distracted by the pretty images. So figure 6 is where they've got the model.
Okay, I'm on my way. I'm here.
Scroll, 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 scroll. Figure
Scrolling?
6H and figure 6A is what it looks like.
Well, let's focus on figure 6A first.
What the fuck is an ostium?
Ostium is
Ost is like
it's
bone, right?
well, yeah, it's typically used for bone in Latin and with medicine, but like osteosarx would be bone cancer. But in this sense, the ostium is the little pore that forms at the surface of the skin that will eventually be the top of the shaft.
Oh, and it does look like it
And my wife
almost
is
like
laughing in the background because I said shaft.
it looks like, yeah, the ostium only emerges at pseudotime like 1.0.
Is that what this figure is saying? No ostium
Because
at
like they get
pseudotime
like a little
0.82.
circle.
And then, yeah, so it's at the last, like, part of development is when the ostium forms, which is interesting.
But like there's some sort of like involution already. Because if you took like figure 6H,
Okay.
so the HF super, so the gray is the skin, and
Yeah.
then oh wait, is like the gray just like a layer of a certain type of cell that's sort of like defining the boundary of the hair follicle, and then within that boundary of hair follicle is
Yes.
where the cells actually start to organize into the different compartments,
Correct.
and the ostium only sort of ruptures through this soup of cells. Oh.
Yes.
Oh my. Because if we look at the iris cone apex, yeah, the ostium, they've only got occurring in the later stages, the left.
Yeah, but you can see where the genes for the ostium are being expressed, and they're coming from the top down.
It looks like they sort of form like a vertical shaft that then sort of segments into like a cone.
Yep.
That cone then gets penetrated by no, the IRS first penetrates the CP layer, and then the IRS penetrates the supra cone, which eventually becomes the ostium.
Yeah, so the super comes from the top down, being expressed from where the pore is going to form, and then it meets eventually with the cavities, and that pushes all the way up to the top, and then it becomes the ostium at the top once it's finished.
Mm,
Interesting stuff.
okay. Yeah.
And knowing
seeing
this
it now.
can help us understand, like, people that can't grow hair. Obviously, you talked about balding and alopecia, but there's other things that, cause issues in growing hair. Maybe this can lead to different discoveries of nullifying the hair loss that is associated with chemo.
Maybe this is saying, are you familiar with like microneedling?
Yes.
Maybe this is saying microneedling doesn't work. Or like organogenesis doesn't require you to punch like a hole in your skin. Maybe the way like microneedling works is that you're like agitating the skin, you're encouraging the sort of like deep layer of macrophages where otherwise there might have been a deficit. And it's not necessarily actually the hole itself which is encouraging a hair to grow out of, but like just like the agitation of the area.
Yeah.
That might be it.
I hear you.
Okay.
I hear you.
Because as far as I'm aware, like the evidence on microneedling is very mixed. And more likely than not, it gives you like acne and like an infection, as far as I'm aware.
I've never had it done, so I can't tell you, but I do know what it is.
Okay, yeah.
All
So
right.
what goes wrong in the development of this organ? Our 4D map of Our 4D map.
Our 4D map.
Alright, let's continue. Sorry. Of normal helical follicle organogenesis provides a foundation for investigating its congenital abnormalities. No,
Congenital.
anomalies.
Read it. Read it. Read.
Sorry. I didn't. Congenital anomalies. I really wanted to like see what that word was, and it was not what it actually was.
Congenial. No.
So they focused on FOXN1 deficiency, which causes a nude phenotype in both mice and humans. So you're right. This might be good for Jada Smith. Despite this dramatic phenotype, hair follicles in FOXN1 knockout mice appear grossly normal at birth and even produce hair, yet hair largely fails to emerge at the skin surface. This failure has been attributed to abnormal ultrastructural organization of the iris and hair shaft in the mature follicle, where FOXN1 is highly expressed. However, whether earlier stages of hair follicle formation are affected remains unclear. So let's get some of the highlights from the results here. So, whole organ pseudotyping calculated jointly for wild type and knockout mice. They ordered all of them, they found the correlations, and they revealed that the knockout follicles follow the same developmental trajectory as wild type mice. However, a key difference emerged. While average follicle length was similar between knockout and wild type, average developmental pseudotime was 21% slower in knockouts than in wild type. They were developmentally delayed. When comparing across similar lengths, knockout follicles were 8% behind in pseudotime. What the fuck? They actually found a significance value for that? Linear mixed model, p-value less than 0.01. All right. So the FOXN1 knockout follicles keep pace in physical growth but lag in molecular progression. Interesting. So the cells continue to divide on pace, but they're not unlocking the proper sets of genes, is what it seems like.
Okay.
As they are hitting the right times. It's like a guy who's like 6'3" and in like 6th grade because he got held back so long. You know?
I mean,
Thankfully you're like 5'8", so I couldn't be talking about you.
I'm not 5'8". What the fuck?
Aren't you like a squid? You're like 5 inches long? 5 centimeters and 8 millimeters long?
No, I'm a large squid. I'm like 4 feet long.
Okay. He's a big squid.
Big squid.
So despite these global differences, the milestones of hair follicle organogenesis remained intact in the FOXN1 knockout follicles. Stem cell establishment and stratification during the first molecular stage appeared normal. The emergence, order, and timing of hair follicle structures, including the hair shaft during the second molecular stage, were indistinguishable between wild type and the knockout. Hair canal formation in the third molecular stage also appeared unaffected. Thus, while the core organogenesis program is preserved, Its execution is slower and more variable in nude mice. Delayed in the French sense.
Yeah,
We next
their hair gene follicle is just not being expressed correctly. I get it.
I just want my hair follicles to grow on time.
I just
Is
want
that
to
so much to
express
ask?
myself with hair. Why can't I express
Yeah.
myself with hair? Why are you making me nude? God.
We next examined whether structures within the follicle. I mean, there's still like
Ask your
weeks
God why he
research
made
now.
these mice nude. That's the real question of evil.
We next for what's it called? For genomic variety. That's why. In case there's selection pressures. That's why God made them nude.
You could have just
See,
said
no
because we
rebuttal.
need test subjects.
No rebuttal from the soy atheist. Yeah, that is
The
exactly
soy
what God did.
theist?
Yeah, that's you.
That's my next
Okay.
nickname. I'm getting rid of salty squid. I'm gonna become the soy theist.
The salty soy theist.
Salty soy theist.
That's pretty good. Okay. So they next examined whether structures within the follicle are affected. They found growth correlations among follicle structures were reduced in the knockout. What does that mean? What are a growth correlation? Because like the structures were the same. That's what they just said. What's a follicle growth correlation? Okay, so they're saying suggesting reduced growth coordination among subtypes. Analysis of, and this is figure 7G. 7G.
I'm on
Number of
my
subtype pairs.
way. I'm on my way. I'm there.
Uh, change in Pearson correlation between knockout and wild type, but this looks like they've got a bimodal distribution. What is it so hard for you guys to plot the Pearson R for the knockout mice in red and the Pearson R for the wild type in blue so that we can take a look at the difference for ourselves? They can't because they hate us. Okay. Next thought is hitting figure 7H. Analysis of keratinocyte subtype abundances over pseudotime revealed altered dynamics in knockout follicles. So lower bulge had greater
Wait,
enrichment
is that not
for.
the bisexual flag?
It does kind of look like it. It's the transhumanist bisexual flag for baldies.
Yes.
Bald bisexual San Francisco
I say
tech
we
pro.
adopt it, we slap some Twin Beaks marketing on it, and we sell them bitches.
We could, yeah. Do you want to keep like the p-value asterisks on the right side?
Yeah, absolutely, just to confuse people.
Okay, got it.
It is
So
red, white, and blue, so it's patriotic. We can sell that.
we are probably the most
We're the
patriotic,
most
quote unquote,
patriotic
wing podcast out there.
animals/scientists ever, probably.
We love our country and it can do no wrong. And even if it can do
We're the
wrong, we're gonna
greatest
tell it
country. We're so
gently.
big.
We're gonna tell it so gently. We're gonna gentle parent our country, just like we need to gentle parent our lead-addled rulers.
Lead-addled?
Because the median age of Congress is.
Oh, because they were licking paint.
Yeah, that was sort of their big thing, Like, in the 70s, they were like, oh, look at all this awesome chemistry we can do. Oh, it made everybody bald and
Mom just
stupid.
painted the house, and the schnozberries taste like schnozberries.
And they can't smell, they can't taste because of COVID, the lab leak from China, by
Fucking
the way. 100% true.
huffing
Confirmed.
brake dust and all that shit off of cars, and they're leaded into gasoline. Is that what you mean?
Oh, back when cars were actually made of metal
When they were made of
instead
real
of
materials and not a
plastic.
supercomputer?
Yeah, when you actually had to drive it instead of the supercomputer cucking
Back
you at every angle.
back when you could fix your own car and work on it in your driveway and order parts with a reasonable price
Back.
instead of,
Oh my god.
being cucked by corporations to having to take your car in to get it fixed for thousands of dollars. Good times. I miss them.
I feel like I'm losing hair. Like this is why nobody likes the left. Maybe wait,
Because
hold
we're
on. Maybe we
correct
are.
about everything horrible happening?
There's no joy in being correct. Can we be honest here? There's no joy in being correct.
That's true. That's why I gave up quanting and just became insane.
Continuing on with our insane quanting, the best of both worlds. So they say, though most trends were subtle and did not reach statistical significance, they've only got two little bits of little nubbins of statistical significance. When using a stringent generalization additive mixed model, when they applied GAM.
Gam?
However, when they applied a difference, the two cell types showed clear differences. Which two cell types? Keratinocyte subtypes. First, while hair shaft emerged at normal pseudotime, its growth is diminished after pseudotime 0.8 in knockout mice, resulting in approximately 26% fewer hair shaft cells by pseudotime 1. That's figure 7I. So, blue, they seem to out-accelerate the nude mice. But I don't know, like, is that enough for you to like lose all your hair? For real? I'm not convinced that they've got a great model that's showing us why these nude mice are not expressing hairs. Or like they're expressing hairs, but it falls out at a particular time.
Yeah, it's also showing in figure J that the upper
The
bulge
upper
is
bulge.
forming
Yeah, of course you
more.
want to talk about
I love talking about the upper bulge.
that. And by that you mean your donut brain that is literally pushing your skin, literally deforming your skin from underneath the way
Well,
it's expanding.
I might have ate something a little too big, bit off a little more than I could chew earlier, and so now I'm trying to digest that through my brain hole, and it's causing a little bit of pressure.
So they suggest the upper bulge keratinocytes are expanded by 18% in the FOXN1 knockout follicles. And interestingly, this expansion occurs in a structure where FOXN1 is not highly expressed, suggesting that the phenotype may result from a bottleneck effect in further differentiation from these cells, or from neighboring cells even, they don't even suggest, or from epigenetic memory from expression in earlier progenitor cells, or cell extrinsic effects. Okay, I guess they do mention it. Never mind. I was wrong.
Whoa, you admitted to be wrong live on 4K? I love it. You should do that
Continuing
more
on.
often.
Nope.
You saw
Well,
it. You saw it happen.
I'm literally gonna read the last like two sentences of this meaty. Now we're done with the potatoes. We've just got meat now. While the strongest effects were again in FOXN1 expressing keratinocytes, other cell types also showed changes in signaling genes. It's more supplemental figures. These observations suggest that the signaling environment is broadly altered in nude follicles. I mean, yeah, all of the cells are depleted of FOXN1. Okay, yeah, you're gonna have a couple of problems expressing your genes. But like, if they could have done some like cell-cell communication analysis on top of this, that would have been great. Like, fuck the pseudotimes, fuck this log2 enrichment shit, fuck looking at these particular like keratinocytes. Show me the cell-cell interactions between the normal hair shaft development and the abnormal hair shaft development. And that's that. That's pretty much the paper because we get to discussion afterwards.
Yep. And that's just them talking about how their system of 4D mapping can be used on other organs and what they can do in the future, and then the limitations of the study.
Yeah, we're not really interested in that. We're not
I hate
interested in those other.
limitations. I don't believe in limitations, actually.
That's so true. I think that all journals should treat every paper like they've got an infinite amount of money and you can just like ask the lab an infinite number of questions to
Daddy, more
dispose of
money,
your infinite
please.
money.
I have questions. I have questions that need answering.
Okay. What do you got? I don't think I'm going to be able to answer them.
No, in a study, you can't answer shit. We need to run a full study to answer these questions.
Oh, okay.
Where are we getting the funding for this?
Can we address that like tomorrow? I feel like if I don't get sleep, my hair
Listen, if
follicles are gonna
you
start
would
falling
like
out.
me and Dispel to fund a study, uh, please like and subscribe so that we can get more moolah and build a research facility together.
We're going
What
to
if we cured
research whether or
cancer?
not what we might be able to prevent cancer,
You never know.
because I don't think we can cure cancer.
Twin
It's
Peaks
like not possible.
Foundation. coming
That
2032.
would be so cool. Yeah.
Thanks for joining us on Beak to Beak. It's been quite enjoyable discussing hair follicle organogenesis and 3D technology here with the pigeon. Please like and subscribe. Well, listen, bitch, I get to pick the article next week, which means it's going to be something insane and it's going to be a
I'm done
good
thinking.
time.
Yippee!
Good. We're taking a
Yeah,
break
no.
from the science. We're going to go explore something else.
We're gonna explore the political science.
I'm wrapping my tentacle around you and I'm dragging you down into the inky depths to pull something dark and hideous up from the briny depths and examine it.
Ew. That's so gross.
I wonder what we're gonna find.
Twin Peaks. You know it's time to get the lab coat on. Twin Peaks. Cause we out here being hella wrong. Twin Peaks. And you know we're never stopping till the hammer drop.
Sorry, hold on. Can we cut that out? That's a bad idea.
Yeah.
Okay. What's on my mind? TV static.